that is the estimated number of plants. And so far, we have 230 species of fungi described. Okay. And we said that 90% of the vascular plants are colonized. Let us just make some small arithmetic and say that means each fungus can colonize 1,304 potential plants. What I did is just divide 300,000 by 230. So we get this much of the partners. That means each fungus can colonize nearly 1,300 plants. And so they are not host specific. Since they are not host specific, they can colonize any plant. And yesterday I told that, but however, they exhibit some host preference. For a particular host, there is always the best fungal partner. And I also mentioned yesterday, they are all obligate symbionts. So we cannot grow them in the laboratory medium. We usually maintain them as pot cultures with the host plant. Next important point is they are ubiquitous. That means they are present everywhere. Temperate country, tropical country, hot desert, or a very moist environment, aquatic place, they are present everywhere. All the continents in the world has reported mycorrhizal fungi. So how is it possible? They are present everywhere in the world, in all soils. So the ecologists have given a very nice reasoning for this. What they have said, these fungi existed in Gondwana land. What is Gondwana land? We know that now we have different continents, Australia, Africa, Asia, and so on. But there was a time when all the land were all together. They were all together. And they later on, they disintegrated into different continents. So this land which was together is called as Gondwana land. So what the ecologists have predicted is that the mycorrhizal fungi existed in Gondwana land when the continental drift took place and the various continents like Africa, South America, Antarctica, all these Eurasia, Europe, and happened. So they went along with the land. So that is the reason why they are present everywhere. They are ubiquitous. So this is the reason given by the ecologist. And yesterday I told about they colonize the roots and they produce these structures like vesicles. In case some people have joined today, I thought probably just 1 or 2 minutes I can spend on this. They produce vesicles inside the root system, and there are about 230 described species of fungi. And these are the arbuscules which they produce inside the root. And these are the 4 important genera of fungi found throughout the world. And of the 4, the most common is A, that is glomus. Any soil anywhere in the world will definitely harbor glomus. That too, they are so common. And inoculation with an efficient mycorrhizal fungus improves plant growth. That also we discussed in detail yesterday. And what is the mechanism by which they improve plant growth? Also, I told yesterday, by increased nutrient uptake, especially immobile nutrients, synergistic interaction with beneficial soil organisms like nitrogen fixers, PGPRs, and so on, protect the plants against soil-borne pathogens, protect the plants against water stress, and they also produce growth-promoting substances like cytokinins, indolestic acid, and so on. So it should be a cumulative effect of all these. It makes the plants to grow better. Okay, now with this background of fungi, let us come to the ecology work that has been done. The earlier workers, both from United States as well as from United Kingdom, these in all the reports, all the reviews, they mentioned mycorrhiza will not colonize aquatic plants. This is a statement given including the mother of mycorrhiza research, Dr. Barbara Mose. So out of curiosity, I told my students, there is a lake very close to the university called as Hebbal Lake. I told the students, why don't you go to the Hebbal Lake, collect the plants, whatever they are, bring it, let us see whether they are mycorrhizal. So they brought 27 different types of aquatic plants. Of course, we have a very good botany department, and they could identify all the 27 aquatic plants. And to our surprise, we found 3 of them were colonized by mycorrhizal fungi. And they are these: Cynotus cristata, Salvinia cuculata, and Icania crassipus, water hyacinth. So we were surprised. I couldn't really, to be honest, didn't believe the students. So I said, I will come with you next time. We will go and collect the plants myself. So I went to the lake, collected these 3 plants, aquatic plants, brought it to the lab, stained it, and saw they had beautiful vesicles and beautiful arbuscules. So we couldn't believe ourselves. Anyway, the paper was sent for publication, and it went for refereeing, and it went to Dr. Barbara Mose for refereeing, and she wrote to me and then asked, only thing doubt I have you collected it from aquatic environment? I said, ma'am, I have collected it from aquatic environment. I have collected it myself. So this immediately it was not published. It was going, there was a delay. And finally, it was published. So it became the world first report that aquatic plants are also can be colonized by mycorrhizal fungi. And these plants, what the 3 plants, what we are describing, they are free floating aquatic plants. That means the plants are above the water. The root system is below. The stem and root system is in the water. So these are free floating aquatic plants. Okay, next to this, I had a chance to go and spend some time in New Zealand. I went there as a visiting scientist for 3 years. So I started working in the mycorrhizal lab. The next lab was aquatic botany lab. So the one professor was in charge of the aquatic botany, Dr. John Clayton. So what he had was nice small cement tanks in which he had the various aquatic plants collected from ocean as well as the large lakes in New Zealand. And any visitor comes, he would just show them, these are the aquatic plants. And I told Dr. John, can I collect your plants and then see whether they are mycorrhizal? Then he said, no, I have already checked it and they are not mycorrhizal. Then he said, okay, you have checked it, but will you allow me to take some plant samples and check? He said, okay, go ahead and then check. And when we checked, these are the 2 plants, Myriophyllum triphyllum and Ranunculus rivularis. These are submerged aquatic plants. That means the whole plant is below about 3 meters below the water level. That's what they call as submerged aquatic plants. So these 2 plants were beautifully colonized by mycorrhizal fungi. I showed John, see, your plants, 2 of them are beautifully colonized. He said, we have tested it so many times, you must have green fingers for mycorrhiza. That is why it's showing mycorrhizal association. Then he said, okay, this is what we have kept as specimens in the institute. Let us go to the ocean or the lake and then collect these plants. So he used to take me in the boat, and I used to sit in the boat because I can't dive. So he used to dive and then collect the plants and then bring it up. So he brought it, again we stained, and it was beautifully colonized. Again, it became the world first report that submerged aquatic plants can also be mycorrhizal. And one thing, New Zealanders went one step further. They said, if that is the case, what the plants they keep in the aquarium along with the fish. So they said you can inoculate the aquarium plants with mycorrhiza so that it grows better. So it went to that extent. Okay. Then another thing which we did was, the report from the United States as well as UK, they all said the mycorrhizal fungi, they sporulate in summer. So out of curiosity, I told my students, every month collect the sample and the soil sample and let us see the spore numbers. Okay. And to our surprise, the maximum spore numbers were obtained in winter, just opposite to what the report that came from temperate countries. Both of us are correct because their summer temperature is something equivalent to our winter temperature. So so the temperature in which sporulates is okay. So though it is different in temperate and tropical countries. And then we collected this the sample, and then how long the mycorrhiza can survive in a sample. So that is what we looked. So what we collected the soil sample, and then it was rich. We knew beginning what was the spore number, and every month we were monitoring the spore number, keeping it at 3 different temperatures. We kept one set at 4 degrees in the refrigerator, another set at room temperature around 25, another at 40 degrees Celsius. We were surprised to see they survived even for nearly 2 years, 24 months. They were the infective propagule numbers were something up to 10 to the power of 3 to 4. So that very clearly showed there is no need for refrigerating or cold room for storing mycorrhizal fungi. You can keep it very well in the room temperature for nearly 2 years. And another experiment, if they are present everywhere, they are ubiquitous. So the ecologists have said they must have existed in Gondwana land, and that is how they are present everywhere. Then there must be 2 reasons. They must be able to disseminate actively, or passively. So these are the 2 ways by which they disseminate and distribute themselves. So we wanted to test how they distribute actively. So we did a very simple experiment. Next to the university campus, there is a battery company, Amco Battery Company. So they were throwing away these empty boxes freely. So I went and asked, can you give us some boxes? They were so happy. They said, take as much as you want. So I brought these empty battery boxes, and we made them, divided them into three compartments. Okay, these are the 3 compartments, what I am showing here, A, B, and C. And between A and B, we had put the wire mesh. Okay, wire mesh. And in inner to that, we added a nylon mesh, wire mesh and a nylon mesh. The pore size of the nylon mesh was such it will not allow the root hairs to penetrate. It will allow the fungal hyphae to penetrate. That is the size we chose. And similarly, a wire mesh and nylon mesh was placed between the compartment B and C. And what we did, we planted a mycorrhizal host in the compartment A, and we were waiting till and we planted a non-mycorrhizal plant in compartment C. And of course, we covered it with sterile cotton. All care was taken, and it was watered with distilled water, sterile distilled water, and all that care was taken. And we were waiting when this non-mycorrhizal host becomes mycorrhizal. That means the hyphae must travel all the way, and the center compartment is only soil and no plant in it. So it should travel all the way to the C compartment and then make it mycorrhizal. So the result, it took 180 days for the plant in the compartment C to become mycorrhizal. So that means it took 180 days to travel 300 millimeters, because the size was 30 centimeters. 300 millimeters. So 180 divided 300 divided by 180 is 1.166 millimeter per day. That means the hyphae can travel at the rate of 1.66 millimeter per day. That means very slow movement, and it has to spend its own ATP to travel. So it is not the mechanism by which it is found everywhere. We said that active mechanism is not the method by which they are present or they disseminate everywhere. So what is the second option? Second option is passive. They must be transported by some other animals or something, which that is how they move from one place to another. In the campus, we have a lot of earthworms. So probably we thought earthworms may act as disseminators of amphifungi. So this particular earthworm, which was predominant, was Eudrilus eugeniae. And you can see the earthworm cast here. And what we did was we just collected the earthworm, we determined the AMF propagules in earthworm cast at different monthly intervals. Okay. October, and thereby it decreased. So that which very clearly showed that earthworm cast has plenty of mycorrhizal inoculum. That is how we find so much of infective propagules. And because it is present, we collected the earthworm cast and then air dried it and stored at room temperature. And we were determining how long, what is the viability of the mycorrhizal inoculum in the stored earthworm cast. And the results showed that the mycorrhizal fungus remained viable for 11 months, almost 1 year in earthworm cast when it is stored in the room temperature. So as per the literature and also the discussion with the earthworm specialists in the university, we could make out that earthworms bring 2 to 5 centimeters soil to the surface in a decade. It's a very, very slow process. They bring this much soil. But when once it comes to the surface of the soil, it gets disseminated by wind, water, etc. So they can travel a longer distance. So the conclusion we could draw that earthworms serve as very good disseminators of amphifungi. And in the campus, we also have lot of millipedes. The species what is dominant is Melogostreptus nigrolabiatus. So here is the fecal pellet of the millipede. In fact, the fecal pellets of these millipedes look so good, one small children can mistake that is a sweet or something like that. It looks so good. And so we thought, let us look at the AMP propagules in the fecal pellet of this millipede. So what was when we did the analysis, okay, the number of propagules in every experiment, we did the spore numbers and also infective propagules. I am concentrating on infective propagule numbers because that is the most important thing. Everything that can initiate colonization. It contains the Eiffel bits, it contains arbuscules, it contains vesicles, it contains spores. So any of them can initiate colonization. That's why I'm concentrating on the infective propagule numbers compared to the spore numbers. So the AMF propagule numbers in the fecal pellet of the millipede and its viability was tested, and the propagule was plenty of propagules were present in fresh fecal pellets. So we just collected the fresh ones and it had plenty of propagules. To our surprise, after 4 days of storage, they lost their viability. So they survived only for 3 days. 4th day, they were all dead. Okay. So we were wondering how to interpret this result. Such a short duration, they lose their viability. Again, with the discussion with our zoology friends, and we could conclude, and also going through literature, the digestive tract of this millipede has a pH of 4. And also it has enzymes, very rich in enzymes, and which digest the inoculum of the mycorrhizal fungi. So from this we concluded millipedes are not good disseminators of amphifungi. Again, in the campus, we have plenty of earthworm, the termite casts. So the termites, and we know that the termite cast, the tip of it is wet and the rest of it is dry. So that means the termites are still actively building their nest there, termite area. So we collected these the samples from termite area, the dry sample, and also the live wet sample. And, and we did it with two different types of termites. Odontotermis, which builds the termite mound above ground. There are some termites which do not build the mounds above ground. They are the subterranean termites which build their nests below ground. And so we tried it with both uh, the above ground as well as subterranean termites. And and we take the samples, and in fact, this particular sample of Odontotermis in the dry termite area, about from 15 samples, live termite area, 20 samples, you can see the spore numbers. Very good spore numbers. The same thing applied with the Microtermis, which is the subterranean termite. Very good spore numbers. But when we tested the infective propagule numbers, which gives how many of them are living, okay, the spore count, it can be, the spores may be living or dead. So, but infective propagule numbers gives only the viable livings, the propagules. So to our surprise, they were all zero. The spore number was so high, the infective propagule number was zero. But compared to the check soil, which was very close to the termite area, you can see the infective propagule was 200. That again showed that something that is present in termite area that is killing the mycorrhizal fungus. So again, going through the literature and discussion with the specialists, we could say the saliva of termites contain benzoquinol. Again, going through the literature, we could see that benzoquinol kills mycorrhizal fungi. So we could correlate why the mycorrhizal fungi do not survive in the termite area. And of course, though there are plenty of termite areas, they do not serve as the good disseminators of AM fungi. Next, we went to the ants. And we see a lot of ants still in the campus. So we took the sample and then looked for the mycorrhizal association. And they were collected, ant nests were collected from the root zone of Lucina, root zone of Goa, root zone of grass, root zone of agave. So different places so that we can compare, and also compare it with the check soil. And the number of samples are 8 in this case. And look at the infective propagule numbers. The infective propagule numbers were very high in this ant hills. And so again, going through literature, we could see that ants bring annually 0.11 kg to the soil to the surface per meter square. But when once they bring it to the surface, they are subjected to further dispersal by wind, water, etc., etc. So the conclusion is ants serve as very good disseminators of AM fungi. Okay, and in fact, we had a project funded by DST on ecology of AM fungi. All this work was carried out through this particular project work. And there was a research scholar who was working in this particular project. Now he is professor of biotechnology. Okay, and when the scholar was discussing with me in my office room, we could see one wasp coming and building the nest in the roof. And he asked me, sir, will this wasp nest will have mycorrhizal fungi? Then I said, I do not know. You take the sample and look whether it has mycorrhizal fungi. So he took the sample and looked whether it has mycorrhizal fungi. Not only that, from different wasp nests. Okay, we have done it from Scleriform Madras Patnam, Scleriform spinole, Eumenes conica, and Rhynchium species. So the number of samples tested is given here, and the number of samples positive for AFF is also given here. And here the number of infective propagules, you can see in so many of them. Of course, this one we had only 6 samples and it was 0. And we take these 3 into consideration, and we have plenty of infective propagules in the wasp nest. And again, going through literature, we could see that a female wasp can carry 40 milligrams of soil or clay, and she can travel 335 kilometers. That means wasps act as very good disseminators, taking the mycorrhizal fungi to a very long distance. So this is about the dissemination, passive dissemination by various organisms. In fact, when I was in US, I was surprised. People used to take me to the forest for the collection of fungi. And one doctor, Jim Trappy, with whom I was associated in Corvallis, and he used to take me. And I was surprised to see a board in the middle of the forest: do not kill rats. They act as disseminators of fungi. Okay, to that extent, they are conscious about this beneficial organism. Okay, yesterday, one of the participant asked whether it is necessary to inoculate mycorrhizal fungi every season. Okay, we had an experiment in this regard. So this was a pot culture experiment and also field experiment, okay, in microplots. So what we did, we had 20 pots, or same 20 plots, which were uninoculated. And we selected in the university campus, based on our analysis, we found that one particular species does not exist in our university campus. That happened to be Glomus intraradicis. So we used that particular species for this experiment. And so 20 pots which were not inoculated, and 20 pots which were inoculated in 3 seasons. We have 3 seasons. We know the Kharif season, Rabi season, and the summer season. We inoculated all the 3 seasons in 20 pots. And another 20 pots we inoculated in the first season and the second season. We did not inoculate in the third season. And the third set of treatment was where inoculation was done in the first, second, and third season. Of course, crops were raised in all the seasons. And the result. Okay, this is the result. Uninoculated, I said that this particular species does not exist in our university soil, so it was 0. And inoculated with only the first season, the number is 3. Inoculated for first and second season, it was 7. And when it was inoculated for all the seasons, this is the number, which very clearly showed inoculation all the seasons is helpful. This is with a particular species which does not exist. Suppose we would have done the experiment with a species which is existing in our soil, probably the results could be slightly different. But anyway, for annual crops, it may be better, because we are raising different crops, it may be better to inoculate every season. But for a perennial crop, the situation is different. When once you inoculate, there's no need for inoculation again, because the fungus is there in the tree species and it can continue till the long time. And here, to support our infective propagule numbers, we also did a little molecular work here. And the spores of mycorrhizal fungi have an enzyme, mallate dehydrogenase. So we isolated this particular enzyme from the spores. And this particular enzyme has 3 isozymes, okay? 3 isozymes. And so we did the polyacrylamide gel electrophoresis, the page. And at the end, you can see when the plants were inoculated with only 1 season, we could not see the bands. When they were inoculated for season 1 and 2, we could not see the isozyme bands. When they were inoculated for seasons 1, 2, and 3, we can see the 3 isozyme bands, which again confirmed that what we got as infective propagule numbers is confirmed by this. And we have a very nice genetics and plant breeding department in the university. So they maintain genotypes of various crop plants. So we just took one particular crop plant, that is cowpea, and they had 101 genotypes of cowpea maintained as in the germplasm bank. So we requested, can we look into these plants for mycorrhizal colonization? They were very happy. So we collected all these root samples of 101 genotypes and looked for the mycorrhizal colonization. There was variation in the percent mycorrhizal colonization. And again, with the help of our geneticist and the plant breeder, we could collaborate with them and come to some conclusion. What were the conclusions? The genetic coefficient variance, they looked into this particular point, genetic coefficient variance. What does this indicate? If the GCV is very high, what does it indicate? It is a character that is used in selection and breeding of crop improvement programs. So in this case, the genetic coefficient variance was very high when we examined all these cowpea genotypes. The second character which the geneticists looked for was the heritability. The heritability, when it is high, what does it indicate? It indicates that it is better for breeding. And the genetic advance is another character which our geneticists looked for, and that was also very high. What does this suggest? That means it is the progeny is better compared to the parents. So because of these three, they said mycorrhizal colonization is not only host dependent, but it is a heritable trait. So it can go from generation to generation. So the plant breeders can look for the varieties which have a higher percentage of mycorrhizal colonization in the breeding programs. So this is the conclusion we could draw from this particular experiment. Okay, and we have done earlier, as I told yesterday, the interaction between mycorrhizal fungi and the rhizobia, the nitrogen fixing organisms, phosphate solubilizing organisms, TGPRs, and so on. And one student of mine was working on soil yeasts. Okay, so we said, why not we see the interaction between mycorrhizal fungi and soil yeasts? And we looked into it, various soil yeasts we tried. And of course, you can see, and people don't work on soil yeast very commonly. And so we can see the Rhodotorula and Metzweikia, Trichosporan, Saccharomyces, Cryptococcus, Debaryomyces. And what we could see is of the various soil yeasts, there was a synergistic interaction between the mycorrhizal fungus and this particular yeast, Rhodotorula mucilaginosa. And you can see the increase in biomass that gave the maximum increase in biomass and the yield of the crop plant. And also the nutrient content of the plant. This experiment was done with cowpea as the host. And so we could say that there could be interaction, positive interaction, a synergistic interaction, not only with rhizobia and phosphate solubilizers, even with the soil yeasts. Okay, and here there's a work that was done by one of the students who came from Mangalore University to do a project work in my lab. And somehow she was very much interested in earthworm coelomic fluid. She said, sir, any experiment you give me, but please include earthworm coelomic fluid. Okay, so we said, okay, let us see the earthworm coelomic fluid, which is called the vermi wash. We know we subject the earthworm, we leave the earthworms in lukewarm water, and they exude their vermi, the components inside the body, and then we again take them back and put in the normal water. And what is collected is the vermi wash or the earthworm coelomic fluid. And what we did was with the rhizobia, actually, in this particular case, the cowpea seeds were soaked in vermi wash for 1 or 2 hours, and they were sown in soil amended with farmyard manure or vermicompost. And the soil, of course, the substrate was amended with vermicompost. The very interesting results, because it was done with the rhizobia, and of course the mycorrhiza also was one of the treatments. And the seed treatment with vermi wash increased the number of nodules and the nitrogen concentration of the plant. This is one thing which we never expected. That is something that is present in vermi wash that encourages nodulation and nitrogen fixation by this legume. And what is the mechanism? Still today, we do not have an answer for it, but it is an area which people can work and then find out what is it that is present in vermi wash, which promotes nodulation and nitrogen fixation by legumes. And coming to a generalization, people ask, which is the most common genus of mycorrhizal fungi present everywhere? The answer is very simple, which I told you earlier also. Glomus is widely distributed throughout the world. Next question is, in tropical soils, which are the predominant mycorrhizal fungi? The answer is Glomus and Gygospora are the ones which are predominant in tropical soils. And in soils, acidic soils, which have a pH less than 5, which is the predominant fungus? Answer is Aculospora laevis. And the soil is very fertile and high in pH, which is the predominant fungus, is Glomus bossei, is the fungus species which predominates. The next thing what we did, being an agricultural university, how the agricultural practices influence mycorrhizal fungi. So we tested various agricultural practices and then what is its influence on mycorrhizal fungi. What are the various agricultural practices we have studied? We have studied the cropping pattern, crop rotation, tillage operation, organic amendment, fertilizer application, crop protection using various fungicides, insecticides, and so on. And how do they affect the mycorrhizal fungi? This is what we studied. And let us go one by one. Here, mixed cropping is a common practice followed by farmers. They either raise a single crop, a monocrop of maize, or a monocrop of soybean. And some farmers go for a mixed crop of maize with soybean. So in this particular experiment, we had the single crop as well as the mixed crop. And we looked for the spore numbers in the root zone soil. And we also estimated the infective propagule numbers in the root zone soil. Okay, so you can see here when maize was raised as a monocrop, the infective propagule was 0.78. Soybean, when it was raised as a monocrop, it was 1. When both of them were cropped together as a mixed crop, you can see it is 2.45. Such a big increase in the infective propagule numbers. So why? Because in a mixed crop, we have two different crops growing closer to each other. So that means the root density is very high. They are taking more nutrients from the soil. And so when the more nutrients are taken, the soil becomes less fertile. And we know the mycorrhizal fungi propagate very well in the less fertile soils. So that could be one of the reasons why we find such high numbers of infective propagules in a mixed cropping system. Again, what we could see when a crop like maize is monocropped year after year for 3 years, many fungi disappear and one particular fungus dominates. And so when a monocropping, this happens with any monocrop, a particular fungus dominates when we take a monocrop. So in case of maize, and it led to the domination of Gygospora gigantea. Coming to another experiment which we did in crop rotation. We know that there are 3 seasons in a year. And in the first season, we took finger millet. Finger millet is ragi, and which is a mycorrhizal host. That means it is very well colonized by mycorrhizal fungi. In the second season, we changed the crop plants. In some treatments, we took the mycorrhizal host, that is cowpea. And another case, we took mustard. Mustard is a non-mycorrhizal host. It is not colonized by mycorrhizal fungi. So that is the host which we introduced in the second season. And another set, this is a field experiment, we left the plots fallow with no crop. And the third season, in all the plots, we raised cowpea, which is a mycorrhizal host. So the first season we had the mycorrhizal host. The third season we had the mycorrhizal host. we changed the crop in the second season. Okay, and we took the spore numbers at the end of the 3 seasons and the infective propagules here. And you can see here, when the mycorrhizal host is grown in all the 3 seasons, the infective propagule number was very high, 2.8. When a non-mycorrhizal host like mustard is introduced, the propagule number comes down. And when the land is left fallow, the propagule number further comes down, which very, very clearly shows that when we leave the land fallow or take a non-mycorrhizal host, the propagule number comes down. And of course, from literature, you can see at least there are 2 cropping seasons to build up the population to the original level. So, and of course, in all the plots, there will be weeds. Weeds also help to build the population of mycorrhizal fungi. So this is the result which we could get from this crop rotation experiment. And alley cropping is another practice which is very commonly followed in African countries. That means they have a few rows of cropland and they have a forestry species, and from which they get the green manure usually and then incorporate into the soil. So this is one of the experiments which we could do in the university campus on alley cropping. And we had 2 different tree species, Lucina leucocephala and Gliricidia sepium. And they were planted at different depth, wide distance between the crops and the tree species. The trees were 4 meters apart, or 8 meters apart, or 12 meters apart. In between, we had the crop plants. So the field crops which we tried in this experiment were either finger millet is ragi, peanut is groundnut, pigeon pea is red gram. Okay, these are the crops which were raised. And these are the 2 forestry species which I told. And we looked for the infective propagule numbers. What is the result we could get? And we took it from all the crop plants, and of the crop species, the finger millet had the highest number of infective propagules. Of the 2 tree species which we tried, Lucina and Gliricidia, Lucina supported higher propagule numbers. And we gave 3 different spacings between the trees, and maximum infective propagules we could get in the crop plant when the trees were planted 12 meters apart. And so this is the conclusion, the last point. Maximum infective propagules occurred in the root zone of finger millet grown between Lucina at a spacing of 12 meters. So that is the conclusion we could draw. And another experiment we had a funding from a Global Environmental Facility. And so this particular, which the funding came from outside, they were very particular about the coffee ecosystem. And this was done in collaboration with the Forestry College in Kodagu district. And so what we did here, we have the coffee is always grown under shade trees. Okay. And so there are 2 types of shade trees, evergreen shade or deciduous shade trees. Evergreen, that means they are always green. The leaves do not shed. Deciduous, at a particular season, they shed their leaves. And again, the farmers there grow coffee under a single shade tree species. Some farmers grow them under a single shade tree species. Some farmers grow them under the 2 or 3 shade trees. And some of them grow more than 3 tree species. And again, there are 2 types of coffee, Arabica coffee or Robusta coffee. So we had a very systematic experiment planned. This was all done in farmers' field. And we took the samples and then looked for the infective propagule numbers of mycorrhizal fungi under the various coffee ecosystems. And what the results we could get, coffee under evergreen ecosystem harbors higher infective propagule numbers. Of the 2 coffee types, Arabica and Robusta, Arabica supported higher infective propagule numbers. And we had 3 different types of shades. So coffee under 2 shade tree species had higher infective propagule numbers. And the least was when the coffee is grown in many shade tree species. So this was a very interesting experiment which the funding agency really liked it. That too because it's an international funding, they liked the results. And another thing which we did was, what is the effect of tillage? And we have the tillage, less tillage, the conventional tillage using bullocks, or deep tillage using the tractor. Okay. And so this here, what is we compared the both. And when we have less tillage, it is better for the fungi. Because it is not disturbing the network of the hyphae. So, and it also helps better crop growth. And, and not only that, when we have less tillage, that next crop also grows better, because the in this particular fungi are not disturbed very much in next less tillage plots. Because I have given the reason, because the tillage disturbs hyphal network, which is important for colonization and nutrient absorption, which is important for better plant growth. And tillage practices, this is not our work, which is I have collected from what others have done. And this conventional tillage, the regular tillage using bullock cart in rice, what they have said it has a positive effect on fungi. The deep tillage has a negative effect on fungi, and the crop they have used in this experiment was rice. And mulching. So between the rows we have a plastic mulching or the straw mulching, whatever can be done. And this is one of the experiments that was done with mulching in wheat crop, and they found it has a positive effect on fungi. Burning the residues, and we know that Punjab, what is happening, all the wheat and the maize residues, and that has a negative effect on fungi. This is the report that came from others' work. And we also tried various organic amendments. So we tried paddy straw, maize straw, pongamia leaf introduced into the soil, compared it with no amendment, and looked into the infective propagule numbers. You can just see that of these, all the straw incorporation had a positive effect on the infective propagule numbers. But of the three straws which we tried, pongamia leaf has the maximum effect on the infective probability, because it is a leguminous crop compared to a cereal crop. And green manuring, again, this was done in a coffee plantation, and in between the coffee plants, different green manure plants are raised, and all of them are legumes. And here we could see that of the three legumes, Dolichos, Cannavallia, and Crotalaria, growing Crotalaria was increased the colonization, but some colonization in the coffee plants. Okay, so because the farmers here, the coffee planters, they use these three plants. That is how we use this, and we found Crotalaria is better in promoting colonization of coffee. And here we have compared farmyard manure with the fertilizer, and it is generally said the fertilizers have an adverse effect on mycorrhizal fungi, whereas the compost or the farmyard manure have a beneficial effect. So we have the infective propagule numbers. You can see the control is 0.65, and it is when we have compost or the farmyard manure, it is 1.2. When we use fertilizer, it came down even less than the control. And we know that in tropical soils are deficient in phosphorus, and when we add phosphatic fertilizer, especially when we add heavy doses of phosphatic fertilizer, it decreases the population, which is very, very well proved. But when we add less less available or slowly available phosphatic fertilizers like rock phosphate or bone meal, it increases the fungal propagules. Similarly, for nitrogen, heavy doses of nitrogen fertilizer also has a negative effect on mycorrhizal propagules. And of nitrate form and ammonical form are the two types which are commonly applied as fertilizer. And what we could see was nitrate is more inhibitory on fungi compared to ammonical form. And in K, not much studies have been done. I could get only one study that has been done in Colombia, and they could see that the level of addition of K fertilizer up to 200 kg per hectare increase the fertilizer, fungal propagules, and also the yield of cassava plant which they used in the study. I think more studies are needed with K fertilizers and its effect on fungi. And again telling the effect of fertilizers, you can use higher dose of 200 kg. It really brings down the spore numbers and also the infective propagules. And in the university, in the agronomy department, they had a very interesting experiment. It is called as long-term fertilizer application experiment. That means they have the treatments, and the same treatment, they'll be applying the same amount of fertilizer. 100% is the recommended fertilizer, 50% is the half of it, 150 is 1.5 times. Year after year to the same plot, they'll be applying the same amount of fertilizer and raising the various crops. So from this, when we took the mycorrhizal propagule numbers, you can again see the adverse effect of using 150% NPK fertilizer. And of course, 50% NPK was beneficial and increasing the propagule numbers. Okay, soil solarization. Soil solarization is done for sterilizing soil. We can sterilize the soil for raising a nursery, for example. One can sterilize the soil by placing a polythene sheet, transparent polythene sheet, and leave it for a few days so that the soil gets sterilized because of the heat. And here we, in this particular experiment, what was done, two different thickness of polythene sheets were tried. One thickness was 0.05 mm thickness, other one was 0.1 mm thickness. And the solarization was done for 20 days, 40 days, and 60 days. So two different thickness of polythene sheets and solarization number of days varied. And so what is the result? We thought the thinner the sheet, probably we will have a better effect. But the opposite happened. When 0.1 mm thickness reduced significantly the propagule numbers to a higher number compared to the thinner thickness of the polythene sheet. And this is one conclusion we could draw. Of course, leaving it for a longer period for solarization naturally will bring down the AMF propagule numbers. So when it was solarized for 60 days, then it brought down the AMF propagule numbers compared to 20 and 40 days. And what is the effect of pesticides? Either we talk of fungicides, insecticides, herbicides, and fumigants. The interesting results, a very beautiful review on this particular topic by Dr. Jim Trappe. And so I could draw some conclusions from his review. And this is the result. Most of the pesticides inhibit fungi. Some of them have no effect. And some of them stimulate the mycorrhizal fungi. We'll just give you examples here. Coming to fungicides, Binomil, thiobenzadole, and triadimenophenone, and pentachlorotriobenzene, they are all inhibitory to fungi. The second group, Captan and Rylan, have no effect on fungi. So it can, when somebody is using fungi, they can as well use this fungicides. Coming to insecticides, Metasistox and Anthrine are inhibitory to fungi, whereas chlorpyrifos and sumicidine have no effect on fungi. And this particular DBCP, 1,2-dibromo-3-chloropropane, stimulates mycorrhizal fungi. It's very interesting report that came that it stimulates mycorrhizal fungi. All the fumigants so far studied are inhibitory to mycorrhizal fungi. Coming to herbicides, glyphosate, ethyl, oxidazole, they are inhibitory to mycorrhizal fungi. 2,4-D, carbendazim, and chlorothalonil have no effect on mycorrhizal fungi. Again, we have another herbicide which promotes or stimulates mycorrhizal fungi. Anilofos promotes mycorrhizal propagules. And looking in acidic soils, one of the methods for reclamation is add lime so that the acidic soils become neutral. So there is an experiment that is people have done applying lime, and reports have come from 3 different countries. It is very interesting. In one country, report says it reduced fungal propagules. Another country report said it had no effect. And the third country report says it increased fungal propagules. So I think maybe the environment, soil type, everything must be different. So probably we should have more studies to draw meaningful conclusion as to application of lime for reclamation of acidic soils. And another study we had, again, this is a project work which was supported by the Global Environmental Facility and the United Nations Environmental Program. And this particular program was done in Western Ghats. And here we studied the diversity of fungi across different land use types in Western Ghats. So in Western Ghats, we have 7 different types of land use. One is the natural forest, which is occurring, and we have the natural grassland and acacia plantations made by man, cardamom plantations, coffee plantations, all man-made, and paddy fields. Okay, so this was the 7 different land types, land use types. And we looked into the spore numbers, infective propagule numbers, and we found that they were the highest in natural forest and grassland, and it was least in paddy fields where the interference by man is more frequent compared to coffee or cardamom plantations. And we could isolate 282 different isolates of fungi in this particular study, and which was identified also. And so the AMF species diversity and richness was also studied. And here the species diversity index, that is the Shannon-Wiener index, which is used for comparing the diversity between various habitats. Okay, the diversity occurring between various habitats. And that also we studied. And we also studied the species richness index, Jack-Sniff's index, which gives the number of species in a community. So both these indices were studied. The results are given in the next table. You can see the diversity index and also the species richness index. And both of them were high or the maximum in natural forests. And it was least in paddy fields. Okay, which very, very clearly showed as we interfere with nature, the propagule numbers come down, and in maximum interference brings down to the maximum extent. And this is an experiment which was done in Arunachal Pradesh, not by us, by some other group. And this is where they have a slash and burn agriculture. They also call it as zoom cultivation or zoom. What they do is they cut the natural forestry species, burn it, and then grow crop plants. So this is what is called as slash and burn agriculture, disturbing a natural forest. Very commonly followed in Northeast, very much in Arunachal Pradesh. So this work was done in Arunachal Pradesh, and the soils were collected from the natural forest and also the zoom cultivated area. Okay, so 44 different species of fungi belonging to 6 genera. I have given the genera: Achilospora, Entrophospora, Gygospora, Scutellospora, Sclerosis were identified from both the systems. But what was the distinct conclusion that came from this study is that for 10 species of fungi, disappeared in the Zoom cultivation, which were present in the natural forest. So by this slash and burn, we are disturbing the diversity of fungi. And of course, the reason is very simple, because of burning, and then we are doing and disturbing the natural ecosystem. This is an interesting study reported from Japan. And what they did in this particular study is, there was a hill actually, and one side of the hill was entering the sea side, and another side of the hill was entering inland. So when there was a seaward slope, in the seaward slope, this particular grass, Elymus mollis, was the predominant grass growing on the hill slope. When the slope was towards the land, the grass was different. It was Miscanthus sinensis was the predominant grass on the hillock, on the land slope. So what they have done, they have collected the roots from these grasses which are growing in the seaward slope and the landward slope. DNA extracted, PCR analysis was done, and they could identify 22 fungi belonging to 5 families. So the conclusion they could draw from this experiment, the diversity of fungi was higher in the seaward slope grass, Elymus mollis, though that had the higher diversity of fungi compared to the grass which was growing towards the landward slope. And of course, there could be many reasons for it. So I personally feel it probably the host preference could be one of the reasons. And of course, the people say that in the seaward slope, there is more soil disturbance. That could be another reason for the more predominance in the diversity of fungi. And what we can conclude from what I have said is, compared to temperate countries, very less studies have been made on the ecology of fungi in tropical countries. And so I think people living in tropical countries, the scientists should study more intensively the ecology of fungi. And it is very well established that fungi improve plant growth. And one of the impediments coming in the way of field application is the fragmentary information we have on the ecology of these fungi. So we have very less information on the ecology, and that is why the application in the field becomes more difficult. And more information is needed on the dissemination and factors influencing diversity. I just gave few examples of how the animals can disseminate fungi. Plenty of studies can be done on the various mechanisms by which fungi gets disseminated in nature. And also I gave examples of very few examples of various agricultural practices, how they influence the mycorrhizal fungi. And there are varieties of agricultural practices which has not been covered. So we can study those agricultural practices and see how they influence the native mycorrhizal fungi. And mycorrhiza workers always advocate two strategies of managing fungi. One is easiest thing, inoculate with the efficient mycorrhizal fungi, which most of us follow, including me. We do that. Inoculate with an efficient mycorrhizal fungi, we get very good response to plant growth. The second category, how we can manage fungi, is managing the indigenous fungi without any inoculation. Managing the indigenous fungi by judicious agricultural practice. I gave some examples which promotes the mycorrhizal fungi. So such studies will help to understand more clearly the ecology of these fungi. Thereby, we can manipulate them for improving the plant productivity without inoculating an outside fungus. So the second category, less studies have been made. So I think more studies are needed in managing the indigenous mycorrhizal fungi for improving plant productivity. I'll stop here, and if there are any questions, I'll be very happy to answer. Can you put it in the chat box also? Yeah. Post your questions. Yeah. Message. So 1 hour 10 minutes.
Thank you, sir.
Thank you.
Thank you, sir, for the informative session. Now the session is open for discussion.
You know,
Session is open to discussion.
Participants can ask your queries in chat box.
Please suggest some books. Somebody has asked here, Dr. Kanta Meena, please suggest some books. Okay, there are a few books which, if you send me one email, I will give you the reply suggesting the books. Sure. Dr. Kanta. Okay. No other questions?
Yeah, any questions from audience?
Yeah, I completed. Yeah.
Yes, Guruji.
There was only one question which I answered. No other questions.
Yeah. Participants, any question from you? Please feel free to ask. Otherwise, you can send an email also to me. Then I can forward to him. If it is not possible.
Okay.
I think there is no questions for us, sir.
Okay.
Yeah. So thank you very much, brother.
Okay, welcome. It's a pleasure.
And I hope all the participants might have enjoyed your lecture. Yes. Thank you, brother. Thank you. Dear participants, please stay alone, and we are going to start next session. Next session will be